Good afternoon. I know I'm between you and lunch, so I'll try to keep it as brief as possible. I know it's a very provocative title, saying we're nailing it, but I think what I'm trying to impress you today is that it's not one thing that is going to solve the validation problem, but we have to look at validation in entirety and how we have to look at validation as a story. And so that's the story I'm going to talk about today. When I was here 2 years ago, I talked a lot about antibody validation according to applications or the functional testing. And we also talked about addressing reproducibility through the various types of methods. Later on, we worked, and Matthias talked about it this morning, is we worked with the IWGAV and established the standards and started testing towards the standards. And then we came up with what we call the two-part validation approach, where we're looking at both specificity as well as functionality of every antibody. So what next, and how can we take this to the next level? And that's what we're going to talk about today. So antibody validation, we talked about applications, we talk about genetic methods, we talk about biological methods of testing an antibody. But all of that doesn't make sense if it isn't in a biology context or the area of biology where the target is relevant. It also is very important to understand the complexity of the target class. So how do we deal with the GPCR versus dealing with an intracellular protein? So today I'm going to walk you through how we at Thermo Fisher are trying to address each of these areas and how we are putting it together as a story in terms of the biology we're trying to address. And so I will start off with the specificity. So back to the IWGAV. So this is our pillars. We saw several pillars before, and primarily taken from the paper that Matthias and team published. And so our focus has been on genetic methods as well as the biological methods. I'm not going to talk a lot about these. It's going to be evident from the data that I'm going to be sharing with you. So the themes are already there in the data, and in the interest of time, I'm not going to talk a lot about that. So I'm going to talk about our two main approaches for the genetic methods. And so the first one is obviously the siRNA or RNAi approaches, where we're looking at knockdown of the protein. So we have tested most of our antibodies, whether we develop them or anything that's already existing in our catalog. We go back, we check, and we test them. Here's a classic example, ERK1 and 2. So this is the standard Western blot that you would see as a functional data looking at ERK1 and 2, and you're looking at antibody picking up signal across various different cell lines. And then we did the knockdown using a combination of siRNAs towards both ERK1 and ERK2. And in addition, we also tested them independently, ensuring that it's actually really specific siRNA knockdowns and the antibody is actually specific. So this is just an example, and throughout the data that I'm going to be sharing with you, you will see several either knockdown or knockout approaches. The piece that I'm going to talk to you more about is how we're using the CRISPR-Cas9 technologies to do knockouts, but also looking at how we're looking at a whole pathway or a whole biology by using these knockouts. So here's an example of the TNF receptor pathway where we've knocked down three main nodes, the receptor, TRAD, and IKK alpha. So we ensure that it's knockouts. So here's the TRAD knockout, and we also have here the IKK knockout in a Western blot. But we're actually looking at the biology here. So if you look at the biology over here, we have the expression of NF-kappa B, which is downstream here, which is usually localized in the cytoplasm, and when you treat with TNF-alpha, it moves into the nucleus. However, when we look at the different knockouts, you see that that movement of the NF-kappa B is stalled and it doesn't move into the nucleus anymore, and ensuring that not only are we getting antibodies that are specific to the different knockouts that we're looking at, but also downstream in the pathway. And I think I will take this on, where we've actually looked at another example where we've just knocked out the node over here, which is an EGF receptor, and we've actually looked at the entire signaling, and that's shown here. So here you have a Western blot looking at the entire pathway. We're using a very relevant cell model as small cell lung carcinoma cell line, where EGFR is known to play a huge role. Here is the controls, the Cas9 controls. So if you look, this is EGFR completely knocked out in the knockout. This is phospho-EGFR, where obviously you don't get any phosphorylation because there's no protein. And this is the phosphorylated form. So this is a combination, so you're seeing a more orangish color for in the controls, but not in the knockouts. But then we looked at all the antibodies that are downstream as well. So if you look at downstream at the MECs, total MEC, you see total MEC because it's an EGFR knockout, but you don't see the phosphorylation anymore. And this is phosphorylation in addition with and without treatment with EGF. So in this data, we're actually looking at multiple pillars within the same blot, and we're able to test multiple antibodies throughout the pathway, and also displaying the biology, saying that here you've knocked out the EGFR, and what happens downstream? What happens to the cells? And so in this way, we're actually validating the entire pathway and also all the antibodies that we have in our catalogs towards this. And so we have, I just illustrated MEC, but it's all the way down to ERK that you can see down here. And in each and every case, we've used a combination of the phosphorylation and the totals. We've also used CRISPR-Cas9 knockouts to delineate antibodies. So the whole validation is also to find, like Matthias had said in his talk, the good and the bad, right? And hopefully not the ugly. And we have here are two different EGFR antibodies. We're doing immunocytochemistry. And we have beautiful staining in the membrane. And actually, this looks really beautiful. But we did the knockouts, and we have very nice knockout, and we have very clear abrogation of signal. And when we did the same knockout for this antibody, we do not see abrogation of signal. So we're able to really call out the bad guys and make sure that we're actually communicating that we take this off our catalog, or if it's a benchmarking, then we are aware that it's not a specific antibody. Similarly, we have another example with the NF-kappa B, and here's again a bunch of different antibodies, and some that work and some that don't work, and here clearly we have one that doesn't work. So I think that very efficiently we use the genetic approaches, not just to look at a demonstration of specificity, but to explore biology as well as clean our catalog and to ensure that we go back, and like Beata talked about, we do go back to our catalog, pick out antibodies and test them in new biology, but also to ensure that we have specific antibodies or not. Moving on to the application of fit-for-purpose applications for the different antibodies that we have and for the proteins that we are studying, I think it's very important to test the antibodies in the right application and in the right methods. And I'm not talking about just IHC or ICC or Western blot, but also within the Western blot, the biology that we need to address. And so here's a classic example of a cell cycle protein. It's an anti-apoptotic gene, survivin, and we can actually do a very simple Western blot. It's ubiquitous. You'll get beautiful looking Westerns. But the idea is to really see how do we understand its biology. And here's a beautiful ICC of the cell cycle, and we know that survivin is specifically expressed in the G2M part of the cell cycle. So you see beautiful expression throughout the cell cycle at the various stages, and clearly demonstrating the potential of this antibody. Here's another example, very traditional AKT1, again with and without treatment with EGF or PDGF and with inhibition. So again, biological pillars, but also addressing a specific application because it's a phosphorylated form of AKT1 and we need to study it in a Western blot. And here again, we have a U87MG as a control because it's a P10 negative cell line where AKT1 is highly phosphorylated. So again, ensuring that the biology is understood in different applications. And I'm going to talk about further, getting more detailed about it. Here is immunofluorescence using retina for optineurin, which is expressed in the ganglion cells in the retina. And here's an example of VAMP1, which is a vesicular protein. It's part of the SNARE family, and it's localized in the hippocampal regions of the brain. So again, different applications and how we address it based on the application. Here is something that was just talked about in the last talk, RNA binding proteins. So we have IGF-2B1, which is an insulin-like growth factor, mRNA binding protein. And this is classic validation. You have expression in certain cell lines which are more like HepG2 and PANC1, but not in others. So I could say we have relative expression in a Western blot. I could say we have relative expression in immunocytochemistry, but it's an RNA binding protein. So we went ahead and we did RNA immunoprecipitation. So we did RNA immunoprecipitation with 3 different pull downs, and we have enrichment here with the IGF-2, but not within 18S RNA, and it binds to the target RNA because it preferentially binds mRNA. So again, ensuring that we're actually addressing the right application for the target that we're studying. Here is chromatin IP. We do chromatin IP a lot for epigenetic targets. But chromatin IP can be done in several different ways, and I think one of the main things that I want to call out in this application is that how do we design our primers in order for looking at enrichment? What are the loci that we're really looking at? So if I take this example of the K36ME3, we know that it binds to the 3' region of active genes as opposed to the 5' end. So we actually we're looking at 2 active genes, GAPTH and beta-actin, and then we have 2 transcriptionally silent loci as controls. But within the genes, we're looking at the upstream region, the promoter region, the exonic region, and the downstream region. And you can really see that there is fold enrichment specifically in the downstream and the exonic regions, but not in the upstream or promoter regions. So it's actually within the same gene we're designing and not really just saying this is active and this is inactive. And I think that's the level of detail we get into, so thereby we demonstrate specificity as well within the same application and within the area of biology. Here's another application, which is flow cytometry. And we have, CD3 antibody. And again, here it is stimulation of, this is peripheral blood mononuclear cells or primary cells stimulated with cytokines, and then asking whether we're getting CD3 picking up. And we can clearly see that it does. And we do a co-stain as a control, which is TNF alpha in this case, and IL-2 in this case. So demonstration again of our specificity of the antibody as well as relevance in the application and using the right cellular environments. So I think what I was trying to impress upon you here is this addresses everything that we talk about in our pillars, the standards, and we're ensuring specificity, ensuring function. But then how do we look at biology? So some of the biologies maybe is easier than others. So for example, I could do the beautiful CRISPR pathway in a cancer cell line, which is more amenable to working with than maybe in more difficult cells to work with. So how do we address challenges such as those? And here is an example that I'm going to talk to you about, is looking at something that's important in stem cells. So here we're looking at a SOX9 antibody, and we have beautiful data here. So we have expression in immunocytochemistry, in Western blot, we have knockdown, and so we know it's functional and it's specific. But we know that SOX9 is important in the formation of the germ layers, so ESC differentiation into the germinal lineages. So we actually took iPSCs and made them into embryoid bodies, and then looked at SOX9 expression in the embryoid bodies, and we're clearly seeing beautiful expression within the germ layers. So we're taking it one step further. We know the antibody is specific, but then we want to make sure that it actually works where it is supposed to work to, because most people who will be using this antibody is most likely going to be studying it in stem cells. So it's important for us to show that it works in that environment. More stem cells is, so we've also tested a bunch of the antibodies that we have. Here's another example of taking iPSCs and differentiating them into multiple different lineages, and then testing different antibodies that we have to these different lineages. So here is an example of optineurin. You saw the IF before. So here it is in the retinal ganglion cells. Here's beta-3 tubulin in neuronal cells. Here is alpha-actin and troponin cardiomyocytes, and here is FOXA2 and insulin in pancreatic cells. So again, we don't just stop with this. We also have tested them for advanced verification, and so here's optineurin knockout in ensuring that this antibody is actually a very highly specific antibody. So multiple pillars that we're addressing to ensure the antibody specificity. Here's another example. In this case, it's a mesenchymal cell line marker. So RUNX2 is a marker that is specific to osteoblasts, and it's very important during osteoblast differentiation. But it is a very transient protein. It's expressed in early osteoblast differentiation, but not in fully differentiated cells. And so that is what is seen here in terms of a time course. So you have in undifferentiated cells, you don't see any expression, whereas in differentiated cells, you see very clear expression during day 7, which is then lost in day 14. So clearly demonstrating specificity, but in the right biological environment. So that was stem cells. Another area that we have, that I'm going to talk about today as an example, is how do we do validation approaches for epigenetic targets, right? So we have the functional validation, the verification with our genetic methods, but then how do we address studying proteins that are important in epigenetics? So we do CHIP, which I talked to you about, RNA immunoprecipitation, and we also demonstrate specificity, especially for the post-translational modifications in this area with either peptide arrays and another method I would call out now, and also using ChIP-seq and ChIP-Western for DNA binding proteins, which we don't have clear loci identified. So here is an example of acetyl histone 3, where we've done your Western blot, your immunocytochemistry, and IP, CHIP using, again, different primers, either upstream where you don't see much enrichment, but then in the exonic regions you see very high enrichment. And this is the peptide array data where you're looking at specificity, which is seen here, and you see very clear specificity to the acetyl modification. So here's two other examples where we've actually benchmarked with antibodies that are either specific or non-specific, and you can clearly see very high specificity for this. Between these two antibodies, you clearly see very high specificity for this one, while similarly for this H3K9AC, we see very high specificity in this antibody, but not in this one. So, and all of this is peptide array, and then we correlate it with the chromatin IPs as well. So again, just bringing everything together. But we also have a collaboration where when we're doing peptide arrays, we're actually looking at proteins in denatured state. And it might not necessarily be as clear or as representative as it is when you have modifications in nucleosomes within a cell. So to address that, we actually have a collaboration where we're doing this, what we call SNAP chip, where we actually have recombinant nucleosomes that have post-translational modifications on them, and then we do a chromatin IP with that. So here's an example where we have H3K, this is an ME1 modification, and an ME3 here, and we have 16 different nucleosomes that have different, post-translational modifications that have been combined, and then we look at specificity. So in this case, we actually have them, so we're closer to the endogenous expression of these PTMs as opposed to a peptide array where it is more denatured. And we see very clear specificity to these antibodies, sorry. Oops. Okay, so I think that these are different enrichment that you see very high specificity for these. So in all these different ways, we're approaching our target. So we're not getting very specific just with the chip or a peptide array, but actually looking at it holistically. And finally, the only class that is left is the complexity of the target classes. And some of them are harder to validate, and whatever method that you use. And we also use this in our development strategy. So here's an example of another epigenetic target where when we are trying to develop specificity to a particular modification, sometimes it becomes extremely difficult because there's so much homology and it's very difficult to identify. So we use different immunogen and screening tactics to get to the modification that we desire and to get to the specific antibody that we desire. So when I'm talking about target classes, here's an example of a very difficult target class to make antibodies for, GPCRs. And in this case, it is to the DRD1, and it's a dopamine receptor. And in some cases, as much as we like to stay away from overexpression of cells and methods that are not endogenous, sometimes you do have to resort to overexpression because you can't test these otherwise. And here's an example of immunocytochemistry where we know that when this is a model where we've overexpressed DRD1 in HEX cells. This work was done in collaboration with Dr. Scholz in Germany. And so over here you have internalization upon treatment with when dopamine binds to the GPCR. And so here, that's clearly with and without dopamine. You see it out in the membrane. And then upon dopamine, you see internalization of the GPCR. So clearly demonstrating that this antibody is actually binding to the right target. And similarly, this was done with IHC, and you see specific expression in the rat brain striatum and the hypothalamus. And here's the control, and here is the Western blot of these cells. So again, difficult target and in the right biology. Another area that we've been working with very closely with the McGill Institute is to look at various players in the Parkinson's disease. And you have several examples that have been highlighted here. I'm not going to refer to each one of them, but our goal has been to develop very, very specific antibodies to these targets, and we demonstrate specificity by generating knockouts for each one of them. And then our collaborators so this was done in collaboration with the McGill Institute, and they are also going to be using that to understand Parkinson's disease. And here are some examples. Here's RME8. Again, here's specific localization, Western blot, siRNA knockdown, and then knockout, which was done at McGill. So, And more such examples over here. You have ataxin here, which is again an important target, and you see knockdown in both applications. So, and here's again SOD1, beautiful data here in neuronal SHSY5Y cell line. And again, you have scrambled and beautiful complete knockdown with siRNA, and we have knockout data for these as well. Another area, and somebody asked this question this morning, and this was kind of surprising for us, is like there are some essential genes that you can't knock down or knock out. And what do you do then when you have a genetic method that might not work? And I think that this was very interesting because when we started off studying this, when we were trying to put a validation plan for this, we said it's citrate cycle, citrate synthase. It has to be absolutely essential. We will not be able to knock it down. We will not be able to knock it out. So what do? But interestingly, when we played around with the siRNAs, we were able to get really beautiful knockdown. And here's two key players in the TCA cycle, citrate synthase and IDH, and that's shown here. We get very beautiful knockdown with siRNA, and this is in a immunocytochemistry context, and this is in a Western blot context. And again, biology-wise, it's in the right cell model, and overall understanding the whole biology with the specificity. So I'm going to summarize by saying, when we look at antibody validation, we're actually looking at telling a story protein by protein. So when you take a protein, you understand what it is, what type of protein it is, where it's localized, how is it being studied, and then develop a whole story on proving that this antibody works in the way a researcher would be using this antibody for. So with that, I'd open to questions. Thank you.
Depth there. That's great to see. Questions? We can probably accommodate one as we dine for lunch.
How many antibodies do you have in the Thermo catalog? And how many have this kind of data? I know it's a rude question, but it's an important question.
I will leave Matt to talk about the number of antibodies we have in our catalog, but I can talk to you about the fact that every antibody that we are developing goes through this. Every antibody that we have developed already, which was already in our catalog, has been tested in this fashion.
So how many proteins do you cover with this beautiful?
It's a journey. We're on a journey. We have covered over 5,000 antibodies so far in just the testing part, and generating this kind of data. It's something that's a big focus for us, and we are scaling up to get this rapidly covering our entire catalog.